Hello students, welcome back to a new video of Sunanda's tutorial. In this video we will discuss about enzymes. So what are enzymes? Enzymes are basically biocatalysts. They catalyze one process, biological process. It increases the rate of chemical reaction without itself being changed or involved in overall process. So it is a very important molecular device which determines the pattern of chemical reaction or transformation. All the chemical reactions or processes are mediated by any enzyme. Okay, and enzymes have many unique properties which make them unique from others. So at first we will discuss those unique properties or characteristic features. At first, they are proteinaceous in nature. Most enzymes are globular protein made up of long chain of amino acids. So proteins, the amino acids are the building blocks of proteins, and those amino acids folded into specific three-dimensional shape to form globular protein, which we can call as enzymes. But few RNA molecules, also called as ribozymes, they can also act as catalyst. Next, catalytic efficiency. These enzymes can enhance the rate of reaction. That means it accelerates the reactions dramatically, often by millions of times, by lowering the activation energy. Later on we will discuss what is activation energy. For now, it is some amount of energy which is required to start a reaction. Then enzymes are highly specific. Each enzyme has specific region called active site which will bind with the substrate. That means or particular reactant like key, lock and key. As a key is specifically made for a lock, like that an enzyme is specifically manufactured for a specific substrate. Then reusability. Enzymes are not altered or used up during the reactions. They catalyze, means a single enzyme molecule can perform its function over and over again. Okay. Next, sensitivity to temperature and pH. So they are very sensitive to pH or a particular temperature. Extreme heat or improper pH can denature the enzyme, which stop them from doing their work. Then requirement in a small amount. Because they are reusable, only a small amount of enzyme is required to process massive number of substrate molecule. Next, reversibility. Many enzymes catalyzed reactions are reversible, allowing a cell to build up or break down substances depending on metabolic needs. So enzyme catalyzed reactions are reversible. They allow the cells to manufacture something, then to break down something depending on the metabolic need of the cell. Okay. Next, how to name an enzyme. Okay, before that, remember, enzymes can be divided into two general classes. One is simple enzyme, second one is conjugated enzyme. Simple enzyme consists entirely of amino acids, and conjugated enzyme contains a non-protein part called as cofactor, which is required for biological activity. Removal of that cofactor from a conjugated enzyme leaves only the proteinaceous component of the enzyme, which is called as apoenzyme, and which is generally biologically inactive. The complete biologically active conjugated enzyme is called holoenzyme. So these are some terms we have to remember. Next, rules of naming enzymes. So enzymes are named using common names ending with ASES added to the substrate or reaction type. Each enzyme has a unique 4-part enzyme commission number, and these systematic naming rules are governed by IUPAC enzyme nomenclature. And each enzyme has a unique 4-part enzyme commission number. Some older digestive enzymes are there like pepsin, trypsin. They retain historical in suffixes. So what are the common rules? So either the enzyme can be a substrate based, add ACE to the target molecule. Lactase breaks down lactose. It is a very important example of substrate based enzyme naming. Or we can name it on the basis of their action. Add ACE to the type of chemical reaction, oxidase. They catalyzes oxidation. Sometimes they work combinedly. Name the substrate and reaction together. Lactate dehydrogenase. Here the substrate is lactate or lactic acid and the work is dehydrogenation reaction. So dehydrogenase. Next, what is enzyme commission number and how the enzymes are named? Every enzyme has an enzyme commission number with 4 numbers separated by dots. For example, EC1.1.1.1. The first digit is the main reaction class. Second digit is the subclass. Third digit is the subclass. And fourth digit is the serial number of that specific enzyme. Okay. So there are 7 main enzyme classes: oxidoreductase, then transferase, EC3 hydrolase, EC4 lyase, EC5 isomerases, EC6 ligase, EC7 translocases. At first, oxidoreductase. So oxidoreductase simply catalyze the oxidation and reduction reaction, addition and removal of hydrogen ion. So they catalyze redox reaction. So the general formula of this oxidoreductase, A gives rise to, okay. Example: oxidases, dehydrogenases, oxygenases, peroxidase. Next one is EC2, which are transferase. They move functional groups between molecules. Like suppose A have functional group B, they react with C, then that functional group will be transferred to C. Examples: transcarboxylase, they help in the transfer of carboxylate group. Transaminase, transfer amino group. Then kinase, transfer phosphate group from ATP to the substrate. Then phosphorylases, they transfer inorganic phosphate to a substrate. Okay. Next group is hydrolases. They break chemical bond using water. Example: phosphodiesterase, phosphatase, peptidase. And the general reaction: so there is a bond between A and B. Now water molecule is added. Now it can become, the bond is completely broken. Next one is lyase. They cleave the bond without water, oxidation or transfer, often forming double bonds. Okay. And example: aldolase, synthases, dehydratase. Next one is isomerase. They rearrange the atom to form structural isomers. Example: mutase. Okay. Next, ligase, they join two molecules together using the energy from ATP. Okay, here the energy is supplied by the adenosine triphosphate. Generally in books 6 classes are mentioned, but sometimes translocase are also added. And what is translocase? They help in the transportation of molecules or ions across biological membrane. Okay, so these are 7 major classes of enzymes. Now, mechanism of enzyme action. How enzymes do their work? A chemical reaction between two substances occur only when atom or ion or molecule of one collides with an atom, ion, molecules of the other. Only a fraction of the total collisions results in reaction, because usually only a small percentage of molecule interacting have the minimum amount of kinetic energy that a molecule must possess for it to react. So when the reactants collide, they may form intermediate product whose chemical energy is higher than combined chemical energy of reactants. In order for this transition state of reaction to be achieved, some energy must enter to reaction other than chemical energy of that reactant, and the transition state is the one with highest free energy. Okay, and the difference in free energy between transition state and the reactants is called as gives free energy of activation or simply activation energy. The presence of enzyme actually lowers that activation energy. When enzymes are present, the activation energy or the energy which are required to start a process is lowered as compared to when enzyme is absent. So this is how enzyme work, by lowering the activation energy. Next is how enzymes operate. The first step of catalysis is when enzyme binds to the substrate and the compound to be catalyzed. The substrate binds to the active site of an enzyme by multiple weak non-covalent interactions like hydrophobic, hydrogen bond, or any irreversible covalent bonds. The free energy released in the formation of large number of weak interactions between enzyme and the substrate, and those energy is termed as binding energy. Those energy are used to lower the activation energy, and the correct substrate can participate in most or all the interactions with the enzyme and thus maximize the binding energy. Okay, so at first enzyme binds with the substrate, they form enzyme substrate complex. Now enzyme product complex is formed. Now the enzyme is releasing the product. The enzyme is released and it is ready for another reaction, and finally the product is formed. So there are two models explaining the binding process by the enzyme and the substrate. The first model, named as lock and key model, it assumes that there is a high degree of similarity between the shape of substrate and the active site or the binding site on the enzyme, and it is typically similar with the shape of lock and key. Specific key is manufactured for that specific lock, and that's how a specific substrate is made or manufactured for a particular active site or binding site of an enzyme. Next one is induced fit model. This model assumes that proteins have some three-dimensional flexibility. It is a flexible structure And according to this model, induced feed model, the binding of substrate with the enzyme induces conformational change in the enzyme that results in complementary feed once substrate is bound. So the binding site has different three-dimensional shape before the substrate is bound. When substrate is bound, the transition state is subsequently formed. Bonds are rearranged in the transition state. Substrate is bound close to atom with which it has to react. As bonds are broken and new bonds are formed, the substrate is transformed into the product, and the product is finally released from an enzyme. So from starting I am explaining you once again. The binding site has different three-dimensional shape. Before the substrate is bound. It is the point 1. Next, when substrate is bound, the transition state, a transition state is formed at the beginning of the binding, and the bonds are rearranged by looking at the substrate. Okay. In the transition state, the substrate is bound close to the atom with which it has to react. And as the old bonds are broken and new bonds are formed, the substrate is transformed into product. And the product is finally released from an enzyme. This is the case of induced feed model. So they induce the change in the binding site of an enzyme. The substrate modifies the binding site of an enzyme. Okay. Now, what are the catalytic strategies of an enzyme? Enzymes commonly employ one or more than one strategies to catalyze a specific reaction. Among those mechanisms, some are very, very important, like covalent catalysis. The active site contains a reactive nucleophilic group that forms transient, temporary, strong covalent bond with the substrate during the reaction, and the best example of it is chymotrypsin. Next, acid-base catalysis. The amino acid side chains of an enzyme act as proton donor when they behave as acids. Sometimes they behave as acceptor in case of bases, to rapidly shuttle protons, activating nucleophile or electrophiles, or stabilizing unstable charges in transition state. Then catalysis by approximation. The enzyme binds with multiple substrates and brings them together in a precise spatial orientation and close proximity, reducing rotational disorderness or entropy and increasing successful collision rate between the reactants. Next, metal ion catalysis. Some metal cofactors act as Lewis acid. They bind and orient substrates, shield negative charges, or facilitate oxidation reduction, electron transfer. So these enzymes actually require, some enzymes actually require metal ions for their catalytic activity. These enzymes can be called as metalloenzyme. Okay. Metal ions can function catalytically in several ways. One role of metal in metal activated enzymes is to act as electrophilic catalyst, stabilizing the increased electron density or negative charge on reaction intermediate, which can develop during reactions. And these metals also mediate oxidation reduction reaction by reversible change in the metal ions oxidation states. Okay. Next, enzyme regulation. How enzyme regulation, or how enzyme regulate different reactions. Enzyme regulation refers to the mechanism through which a cell controls the activity, abundance and localization of its enzymes. This control is very crucial for maintaining metabolic homeostasis or equilibrium. It also prevents wasteful production of molecules, and it allows the cells to respond rapidly to changing internal as well as external conditions. At first, allosteric regulation. So, by allosteric regulation we can control enzyme. This occurs when regulatory molecule, either activator or inhibitor, binds to an enzyme active site other enzymes at a site other than active site. So enzymes generally have one active site or binding site. So this activator or inhibitor bind at other site other than active site, at a site which is other than active site. That site is known as allosteric site. Allosteric site. They either activate or inhibit a certain process. Then allosteric inhibition. What is inhibition? This binding changes the shape of active site so that the substrate can no longer fit properly and do their work. And what is activation or allosteric activation? This type of binding alters the enzyme shape to increase its affinity towards the substrate and finally speed up the reaction in a positive way. Okay. Then covalent modification. So I have already told you, covalent modification of enzyme involves the reversible or irreversible attachment or removal of chemical groups to alter enzyme's activity. It acts as a key regulatory switch in cellular processes. Types of covalent modification, either it will be a reversible modification or irreversible one. Reversible is fast dynamic change where functional groups are added or removed by specific enzymes to fine tune the metabolic pathways and signaling. Example, phosphorylation, addition of phosphate group from ATP to any other amino acids via protein kinases, reversed by protein phosphatase. It adds a bulky negative charge that shifts enzyme conformation. Okay. Next, irreversible modification. It is permanent chemical change, most notably proteolytic cleavage, which transform inactive precursor protein called zymogen to active enzymes. Some enzymes like trypsinogen are activated in gut via peptide bond cleavage by enteropeptidase, preventing premature tissue damage. So this is irreversible modification. Okay. Next, feedback inhibition. So we will discuss inhibition part in a separate video. For now, what is inhibition? In a process, the product, the excess manufacture of a product or excess release of a product itself inhibit that process. When the product built up in high concentration, it shuts down its own production to avoid waste. Next, proteolytic cleavage. Some enzymes are synthesized in an inactive precursor from inactive precursor form called zymogen or proenzyme. They require specific peptide bond to be cleaved by protease before they become active. And this is very important for digestive enzymes and blood clotting factors, so they do not destroy the tissue that synthesize them. Okay. Next, what is compartmentalization? Cells restrict certain enzyme to specific organelle. This physical separation of enzyme from potential substrate, and they create an optimized microenvironment of that particular enzyme, such as some enzyme require highly acidic pH inside lysosome, and that particular microenvironment is required for the enzyme to function efficiently. So some enzyme require a specific um, environment or conditions to work efficiently, and that environment is supplied by that particular organelle. So that enzyme is specifically found in those particular organelles. Okay. Then enzyme synthesis as well as degradation, a slower but long term regulatory strategy where cell controls the actual amount of enzyme present, and this is managed by altering the gene expression to increase the production by induction or to stop the production by repression of enzymes mRNA. Okay, so there is a genetic control and management system to alter the gene expression to increase the enzyme production when there is a need of enzyme. Either they increase the production by induction or they stop the production by repression of an enzyme's mRNA. Okay. So it is all about your enzymes regulation. In my next video, I will discuss other important parts of an enzyme chapter of CSIR NET Life Science syllabus. So please stay tuned. If you like this video, please hit the like button, share with your other friends, and please subscribe to my channel Sunanda's Tutorial. Remember, this is only the part 1 video of enzyme. In my next video, I will discuss about enzyme kinetics, enzyme inhibition, then different factors affecting enzymes. So please stay tuned. Thank you.